Good morning, my name is Wang from Johns Hopkins University. Today I'm going to talk about my work, mean correlation relationship biases co-expression analysis. Gene co-expression analysis is widely used in the reconstruction of gene regulatory network. A general pipeline is to use gene expression matrix to generate the correlation matrix and then apply network analysis methods such as WGCNA and the graphical lasso to predict the gene regulatory network or gene clusters. There are some previous studies that has found the confounding effect of gene expression level towards co-expression analysis. One study shows that genes with high expression level generally have more co-expression signals than gene with low expression levels. Here the color denotes the co-expression signals. Another study shows that most of the observed differential co-expression can substantially explained by the changes of gene expression levels. However, these studies did not furtherly explore the relationship between gene expression and the co-expression. In our study, we want to explore the dependency between the gene expression level and the correlations. Here we name that as mean correlation relationship. We want to furtherly examine the impact of this relationship on the downstream analysis, and we developed the method to remove this dependency. Firstly, to compare the correlation distributions across genes with different expression levels, we first partition the correlation matrix by equal size bins after sorting the genes by their expression level. Here, each bin corresponds to 2 expression levels. And we firstly looked into those bins on the diagonal where the 2 groups of genes have the same expression level. We compare the distribution of those bins, and we find that the high expression bins have high variance of its correlations distribution. For the entire correlation matrix, we quantify the variance of each bin using interquartile range or IQR. Here is the width of each box, and we find that the increase of expression level of either gene would cause an increase of IQR. We also find the marginal distribution of IQR and the expression level of the lower expression group of each bin, and we also see a positive relationship. We furtherly explore the difference of the correlations distribution across different bins using QQ plot. We use the bin with the second highest expression level on the diagonal as reference, and we compare its distribution with other bins on the diagonal. And we find that their distribution not only differs in scale, but also in the shape. Next, to examine the downstream impact of this mean correlation relationship, there are a number of studies estimated the co-expression genes by setting a threshold. Here we set 0.1% highest correlation as signals in each bin, and we compare their signals distributions across the bins on the diagonal. And we find that the bins with high expression level have stronger co-expression signals than bins with low expression level. For the entire correlation matrix, we set 0.1% highest correlation as signals, and we compare the expression level between signals and all genes, and we see a prediction bias towards high expression genes in the co-expression signals. To check whether this bias comes from the true network, we use the protein-protein interaction database, and we compare the distribution of gene expression levels for those genes corresponding to the interacted protein pairs with all the genes, and we see no bias in the PPI network. To avoid such prediction bias, we developed a method called the spatial quantile normalization to remove the mean correlation relationship. The main idea is to partition the correlation matrix by equal size bins after sorting the genes by their expression level, and then apply one-dimensional quantile normalization in each bin. To smooth the normalization, we estimate the distribution of each bin, here is X, using a larger bin, here is Y, and then apply one-dimensional quantile normalization on S using its estimated distribution. Here the target distribution is the bin with the second highest expression level on the diagonal. We apply this normalization on the entire correlation matrix, and after normalization, the distribution of correlations are approximately the same across all expression levels. Our method used the overlapped bins Y to enforce smoothness of the normalization. While the outer bins Y are overlapped, the inner bins are not, which makes sure that each point is transformed only once. Unlike one-dimensional quantile normalization, our method does not guarantee that the distribution of correlations are exactly the same. Also, our method can be applied to other types of two-dimensional data with order. Here, the data is ordered by the gene expression level. We evaluated the method using the same plots as before, and we find that our method removes the mean correlation relationship and the co-expression bias. We furtherly examine our method using the predicted signals involving transcription factors. As we know, transcription factors are important regulators with low expression level. The plot shows the percentage increase of predicted signals involving transcription factors after applying SPQN, and we find that after normalization, there is an increase between 5 to 20% signals involving transcription factors. There are some studies shows that removing principal components can reduce the impact of battery effects towards the network analysis. Here we removed different numbers of principal components from the data, and we find that the mean correlation relationship exists before removing principal components and still exists after removing more principal components. So this work is supported by Casper Hansen with great help from Stephanie and Leandro. Our work is available on bioRxiv, and our R packages is available on Bioconductor. Thank you.
Wonderful. Thank you for a very interesting talk to start us off. Without further ado, we're going to jump into the next talk by Ting Huang.
Hello everyone, good morning. I'm Ting Huang from Northeastern University. In this presentation, I will talk about MSstatsTMT. MSstatsTMT is an R package which can perform statistical analysis for TMT labeling proteomics experiments. So in TMT experiments, labeling experiments, the peptides from different samples are labeled with isobaric variants of the same mass tag. We also call it channels. And then the labeled samples are combined together to produce a single biological mixture, and the biological mixture will pass by the mass spectrometry at the same time. In this way, TMT can reduce the technical variation between these samples. However, in many investigations, the number of biological samples are often more than the number of channels, which means we need multiple TMT natures for such investigation. And statistical analysis becomes challenging for such large-scale experiments with multiple natures because of the complex experimental design and the missing values across MS runs and the batch effects across MS runs. MSstatsTMT was proposed to solve these problems. Here is the workflow of MSstatsTMT. First of all, at the stage of experimental design, we have many options of allocating samples to natures. We are particularly interested in the experiments where all the samples cannot be fit in one single mixture, which means we need to have multiple mixtures, and these mixtures contain multiple biological replicates. Occasionally, we may also have technical replicate run for the same biological mixture, and this design can be unbalanced, which introduces more challenges for statistical analysis. After we have acquired the raw spectrum based on the experimental design, the next step is data pre-processing, which is very important. Data processing takes the raw spectra as input and output a list of identified PSM with reported ion intensity. This is exactly the input to MSstatsTMT. However, different data processing tools may have quite different output format. In order to help our user use MSstatsTMT provides converters for different data processing tools, such as Protein Discoverer, MaxQuant, OpenMS, SpectrumM. The converters can first generate the required format input for MSstatsTMT, and then the converter can also pre-clean the data. It filters out shared peptides, it aggregates peptide spectral level data to peptide ion level. If there is fractionation, it can also combine fractions. After we have the pre-processed reported ion intensities, next step is really where our statistical work begins. We want to give statistical conclusions based on the reported ion intensity we have. Then the first step is to apply spectral level normalization. Here is the plot we will look at the beginning. Different panels represent different MS run which profile different mixtures. Inside the panel, each box represents one channel. The values inside the box are the reported ion intensities for over all the peptide ions, all the proteins in that channel. We will assume that the total protein abundance are equal across run, across channel, across run. In other words, we assume there's only a small proportion of proteins are changing in abundance. Then we apply the global equal median normalization to the boxes. Now the boxes becomes equal in their medians. Next step is to summarize the reported intensities to protein level intensity. Again, same as before, the panel here means one MS run. And then the data here is for one particular protein. This is the reported ion intensities for one particular protein. The line inside the panel is one peptide ion mapped to that particular protein. So we want to, for each run and each channel, we want to summarize these values into one single value, the black dot here. We use 2K median polish to robustly summarize these points, these values into protein intensity. The 2K median polish is is robust summarization and not affected by outliers. Now we have the protein summaries. But the protein summaries are not comparable across MS runs because we may summarize different peptide ions across different runs. So in next step, we normalize the protein summaries across MS run. Here we introduce a term called reference channel. In TMT experiments, we may have we can have at least one reference channel in each MS run. We expect no biological variation in the reference channel. In this plot, the dots here are the protein summaries, and the blue dots are reference channel, are the protein summary in reference channel. We expect no biological variation in the reference channel across runs. So if we see intensity variation in the reference channel, it reflects technical artifacts. So based on the reference channel, we can normalize the technical artifacts between MS runs. What we do here is we equalize the reference channel across MS run and apply the same shift to the endogenous samples in the same MS run. After the reference channel based normalization, we can see that all the reference channels now are equal across MS runs. So now we have the normalized protein summaries. Next step is to perform statistical modeling and inference. Statistical modeling and the inference is difficult for the large-scale experiments with multiple measures because the design for such large-scale experiments can be complex, and we need to accurately represent the experimental design by fitting a linear model. And we also need to accurately estimate the variance components based on the experimental design. So we developed a linear mixed model which takes into account that we have samples which are mixed in one single meter. We have samples which are acquired in one technical replicator run. We are interested in comparing different conditions. We have different multiple biological subjects. This linear model really represents a general experimental design for TMT experiments. It can be easily adapted to other designs. For example, if we only have single meter, we don't have multiple meter, then we can remove the mixture term from the model. And the last thing that's exactly how MSstatsTMT works in background. MSstatsTMT automatically detect what type of the design the data set has, and automatically fit the counter linear model for the corresponding design. The last thing we do for inference is we apply the entangle-based moderation as that in lemma to our statistical inference. As a summary, MSstatsTMT performs relatively protein quantification, normalization, differential analysis for TMT labeling experiments. It can be applied to experiments with multiple meters, multiple technical replicates runs, and unbalanced data. MSstatsTMT takes directly the output from different data processing tools, and MSstatsTMT was published in Bioconductor in 2018. We also published the manuscript for MSstatsTMT very recently in MSCP. So finally, let me thank my advisor, Professor Vitek, and my colleagues, Mina and Sushun. We developed the tool together. Let me thank my collaborator from Roche, which developed the benchmarking dataset for the MSstatsTMT paper. That's all my presentation. Thank you.
Wonderful. Just a quick reminder to continue to ask questions in the Pathable chat poll. And the next speaker is Simina Boca.
Hello, everybody. I'm very excited to be here today and talk about the swfdr package, and specifically how to use it to estimate false discovery rates conditional on covariates. This is joint work with this wonderful group of people. So the basic problem that we'll be talking about is the issue of multiple testing. Many of you may be very familiar with this issue because it's a very, very common issue in bioinformatics as well as modern science in general. So the problem is that we often need to test to see whether there's an association between hundreds or thousands of variables or features, and oftentimes one single outcome. So these are some examples where this may come up. And so, you may be wondering, if you are familiar with this issue, why do we need more methods to do this? Because we already have a number of methods. The very famous Bonferroni approach to control the family-wise error rate, and then the equally famous Benjamini-Hochberg approach to control the false discovery rate. And Benjamini-Hochberg has been around since 1995. More recently, but still early 2000s, the story or Q-value approach to estimate the FDR has also been developed. And that's actually in the Q-value package, which is another bioconductor package. So why do we need more methods? So the answer to this question is related, surprisingly, to the original Star Trek series. Let me just give you a couple of clips to explain.
Scotty, where's that power? Coming, sir.
Scotty, where's that power? Coming, sir, but he doesn't seem totally convinced.
We need more power. We haven't got it.
We need more power, we haven't got it. So basically, we need more power. And so one approach for power, to get more power, is to use an adaptive procedure, which was not available back in the original Star Trek, but we know how to do it now. So the false discovery rate depends on the overall fraction of null hypotheses, which are the variables or features not associated with the outcome. And this is often denoted by pi 0. You can remember this because 0 often stands for null. And there are adaptive FDR procedures, such as the Q-value approach, actually, which incorporate an estimate of the pi0 in estimating the FDR, and that estimate is based on a distribution of p-values. So that's basically related to the magic of empirical Bayes. That ends up being equivalent to empirical Bayes. Now, what if we have other data that we can use in our estimates besides the p-values themselves? What data could those be? We could have external covariates, for example, which are also known as metadata, co-data, or feature-level covariates, depending on what paper you read. And then we can incorporate these into an adaptive procedure and help with the decision of whether or not to reject the null hypothesis. So if we reject a hypothesis, it means that we're actually making a discovery. So we're saying, okay, we think that there is an association here between the feature and the outcome. So there are many examples of these covariates. They depend on the field. We'll be talking about the first one here. I'll be talking about the first one here. It looks at genome-wide association studies. If you're interested in this area, I really recommend that you look at this paper by Ting, Kortauer, et al. That I'll also be describing a little bit more later. So we basically use a regression approach to incorporate covariates. We regress whether or not the hypothesis gets rejected using a logistic regression on the vector of external covariates. And so we use that to estimate pi0 of x. And so pi0 of x, it is this probability of being from the null hypothesis, but conditional on the covariates. And so this becomes a function of the covariates, as does FDR of x. So after we obtain pi0 of x through this regression approach, we can then simply use it as a plug-in estimator for the estimate of FDR of x. We just multiply it by the Benjamini-Hochberg transformed p-value. So that seems pretty simple. It's not too bad once you get this estimate, but it actually works very well in practice. It's essentially equivalent to the story q-values if there are no covariates. The only difference being that if you want to use a logistic approach here, which does work better if you have multiple covariates, it'll be a little bit different from story, because story would be exactly equivalent to using a linear regression approach. You can use either one of them in our package. And then here is the actual publication, which you can take a look at. And then here is our GitHub repository that has the code for our package. And I'll be making the slides available, so you can feel free to use any of these links. I mentioned I'll be focusing on GWAS, or genome-wide association studies. So we consider in our paper and also in our package a meta-analysis from a genome-wide association study for BMI. So that basically means that we have a lot of SNPs, over 2.5 million SNPs, many individuals, and then for each SNP, we look at the association between the SNP and body mass index. And there's over 300,000 individuals considered. Most of them are of European origin. This is a common issue for a lot of these studies, but unfortunately it is the case. And so we focus on the results for the individuals of European origin. One thing that happens for genome-wide association studies that's maybe not as common for other kinds of studies is that different SNPs will be genotyped for different individuals, and that leads to a different sample size per SNP. So the sample size is vary per SNP, and then minor allele frequencies also vary per SNP. So basically those are the frequencies of the least common allele for each SNP. And so that's a little bit different from other studies, but it means that these are also natural external covariates that we can use. The SWFDR package includes a subset of results for the individuals of European origin for 50,000 random SNPs. So Jeff Leake always says, you know, if you can't include all the data, or if you can't plot all the data, just look at a random subset. And the entire data set would have been too big for the package, and also it takes a very long time to run. We do have the code available in supplementary information on GitHub, and also information on how to download the data set if you want to actually reproduce our analysis. But in the package, we only have 50,000 random SNPs. So just, you can load and explore the data set. So this is the library. You can also look at the Q-value package if you want to compare. This is the object. We tried to go for a descriptive name, but then we just changed it to a shorter variable. So you can see it's 50,000 SNPs. And then, it's basically just a tibble, which is just like a data frame, but just a little bit friendlier to use. So for every single nucleotide polymorphism, this is the major allele, the minor allele, the frequency of the minor allele using the HapMap database for populations of European origin. And then these are the estimates from the regression model for each SNP. And then this is the p-value that we focus on. And then we also have n. This is the sample size. And then we have one more variable. We have these bins, basically tertiles for the minor allele frequency. And in the paper, we actually have a model where we look at the tertiles, and then we have splines for n. But in this example, I'll just go for just a simple model where we just put both of these in the regression approach, the regression model. So basically, after loading the data set, you can use the LM_Q-value function, which is based on the Q-value function in the Q-value package, which I mentioned is developed by John Story. And so we can use this. These are just the outcomes. It's like the outcomes, but it's the P-values. And then here we have the matrix that we use for the covariates. And then this is similar to what we get from the Q-value package. So basically, the cumulative number of significant calls using the P-values, we get more than using the Q-values. These are the covariate adjusted Q-values. But of course, we know that using the P-values is not actually the correct thing to do because of multiple testing. And so we still get 254 significant ones at 0.05. This is only about 7 more than using the Q-value package, so we're not gaining like a ton of power. But it is still a little bit. And then also, like I said in the paper, you can actually use a more sophisticated model instead of just putting these in the model directly. So there are a few other things you could do with them. And then you can also explore the estimates of the probability of being null, considered as a function of the covariates. So these are the estimates that we get of pi0 of hat in the histogram. And then the line here is what we would obtain without conditioning, which again is basically what we get from Story. And so this is not in the package right now, but it is in the development version of it, which is available here. There's other plots that Tomasz developed, Konopka developed, that are also found there. And so that we hope to push to very soon to future versions of the package on Bioconductor. Okay. So I just wanted to spend the last minute or so just talking a little bit about the combined powers of open access and open source. And I think it's also a bit of like a story of our persistence for this method, but also how much we were helped by these ideas and by the availability of basically preprints and open source software like Bioconductor, like the availability of putting things on GitHub and so on. So I mentioned, there's this great paper by Kortauer et al. It's really a tour de force. They compared a lot of methods that controlled false discovery rates adjusting for covariates. They were able to compare 8 methods in terms of FDR control power and usability on many simulated and real examples. They only use the univariate covariate. We can use a multivariate one, but some of the methods that they compared only had the possibility for a univariate. Now, while they were doing this, our paper was in the process of what I call like flunking out of a series of journals. So we had a preprint out, which was great. And we had already added the method to the swfdr package, which Jeff and Leah had developed a while earlier. So that was all good. And we actually got a good score in Kortauer et al. So this is their comparison of all of the methods, where the BL method, which I sort of vainly just named Boca leak just because I have a lot of trouble with acronyms. So we did really well, except for actually we only got a half full circle for usability. So a full black circle is the best you can do. So we got black for everything except for half a circle for usability. And that's because you had to go through a couple of different steps. You didn't have the LM_Q, we didn't have the LM_Q value function. At the time, they could just kind of take it all in directly. And so what was great, though, was that Tomasz Konopka, who is in the United Kingdom, so he had read this paper, then he read our preprint, and he offered to help us out with improving the usability aspect. And so he wrote the LMQ value function. He's actually one of the main developers for the swfdr package right now. So I think this is a great success story that indicates that it's really good to have, your code be open source, and then also to have, preprints posted, because even though it can take a while from the time you post a preprint to the time your paper gets published, people can actually learn about your method and then also use your method, and then actually you can get more people to contribute to your project. So that is all I wanted to talk about today. Thank you so much, and I'm excited to take questions.
Wonderful. So 3 great talks, 1 more to go. The final talk in this session is from Philippe Boileau.
And some things do called sparse contrastive principal component analysis, or SCPCA for short, which we use to explore high-dimensional biological data. Before I actually jump into presenting SCPCA, I'll provide some quick motivation as well as some background. So let's start with the motivation. As you all know, a common issue that arises in the analysis of high-dimensional biological data is the teasing out of meaningful signal from technical noise. So in this toy example over here, I've plotted the first 2 principal components. There are 2 distinct biological groups within this embedding, one green and one purple, and each group is split by a batch effect, which which is denoted by the observation's shape. Now, before we go any further in our analysis, we'll have to remove this batch effect. There are plenty of nice methods to do this, and they all work fairly well, but a lot of them make restrictive assumptions about the data generating process. Instead, what we'll have to do, or what we'd like to do at least, is remove this batch effect without making too many untenable assumptions. And this is where SCPCA comes in. Now, SCPCA is really just a combination of two different dimensionality reduction methods, contrastive PCA, which will abbreviate SCPCA, and sparse PCA, or SPCA for short. And I'll briefly introduce these two methods before we go any further. So CPCA, let's start with that. CPCA removes unwanted variation by taking advantage of the data's experimental design. That means, given a data set which contains both the wanted and unwanted variation, which we'll refer to as a target data set, and a data set containing only the unwanted variation, referred to as a background data set, CPCA will deflate the unwanted variation in the target data set. Now, a nice sketch of how this is accomplished is summarized in the first figure of Abid and others, the developers of this original method. Now, the first step is to compute the sample covariance matrices of the target and background data set. Next, we take their difference and multiply the background's sample covariance matrix by a contrastive parameter denoted by alpha. We then take the eigendecomposition of this difference and use its eigenvectors to rotate the target data into a space that hopefully is free of unwanted variation. And this is illustrated in the plot to the right. Now, although this is definitely an interesting approach for the removal of unwanted variation, there are a few issues. Namely, since this method is essentially a PCA but applied to a different kind of covariance matrix, it suffers from the same issues that PCA does in high dimensions. For example, interpreting CPCA's loadings vectors, the vectors that define the linear combination of each of the contrastive principal components, is actually quite difficult since it's filled with plenty of non-zero values. Additionally, these values can be unstable, and so can the principal components. Even worse, though, is the fact that the contrastive parameter, which really drives the ability to remove the unwanted variation, is selected through visual inspection of many CPCA plots. Now, all analyses are subjective to some degree, but I think most people would agree that this is too subjective. So based on at least the first drawback of CPCA, you'll probably see where I'm going with sparse PCA. Now, for those of you who aren't familiar with sparse PCA, it's a method that was originally developed by Zhu and others to increase the interpretability and stability of PCA in high-dimensional setting, which we often encounter in high-dimensional biology. Now, I want to briefly go over how this is accomplished, but I'm not going to go into the math in too great detail. Essentially, what you have to do is reframe PCA as a linear regression problem, and then include an L1 and an L2 penalty in the objective function to sparsify the loading vectors that are produced by PCA. And this is really just analogous to an elastic net regression in a typical regression setting. Now, as I said before, this is really just a simplistic overview of SPCA. It's actually much more complicated than this, but it is important to note that there are a few very efficient implementations of SPCA, such as that recently came out by Erickson and others in January of 2020. And this is one of the methods that we use in our own SCPCA method. All right, so with that said and the background done, we can finally jump into defining what our method is. And as I mentioned before, it's really just a combination of contrastive PCA and sparse PCA with a few minor modifications here and there to make sure that these two algorithms play well together. And the resulting dimensionality reduction method is really just a sparse version of contrastive principal component analysis. That means the contrastive principal components that we produce are now more interpretable and stable at high dimensions. But that's not it. We still have to handle our hyperparameters. In particular, contrastive PCA has the contrastive parameter, and SPCA has an L1 tuning parameter that defines how sparse our loadings are going to be. Now, it's typically not very easy to define an optimal set of hyperparameters in an unsupervised setting, since we don't actually have access to ground truth. But we found that if we were willing to make a few simplifying assumptions about our data generating process, then we could come up with a reasonable heuristic to do just that. Our procedure goes as follows. First, we specify a grid of hyperparameters, and then we apply SCPCA to our data using every possible combination of our L1 and our contrastive parameters. We then apply a clustering algorithm to the resulting embedding and evaluate its clustering performance using the average silhouette width. We'll identify the hyperparameters which maximize the average silhouette width across an embedding as being the optimal set. Now, of course, this procedure only really works if we expect to find distinct clusters in our data. Thankfully, this is often the case, but we found that even if it isn't, this method continues to work quite well. And if ever there's an issue, or you believe there might be an issue with finding non-generalizable patterns in the data, we've also implemented a cross-validation version of this heuristic to try and avoid that kind of situation. So now that you're familiar with this method, we can talk briefly about its implementation. SCPCA is implemented in the SCPCA package, which was first released in Bioconductor 3.10. This package also provides two flavors of CPCA. CPCA with the automated contrastive parameter tuning approach that we discussed in the previous slide, as well as the original approach that uses visual hyperparameter tuning. There are also some relevant details that are worth mentioning. The first is that this hyperparameter tuning framework is embarrassingly parallel. So all the package's methods that rely on it also possess parallel counterparts. These parallel versions rely on the BioC parallel infrastructure. Second, for those of you who might want to apply this method to some single-cell RNA-seq data, the SCPCA package integrates fully with the single-cell experiment container class. And finally, these contrastive methods operate over a covariance matrix-like structure. And that means that the computational complexity grows not with the number of observations, but with the number of features. And so if you plan to analyze any extremely large data sets, you'll actually find these contrastive methods are computationally much quicker than other competing methods like TSNA and UMAP. So with that said, let's take a look at SCPCA in action. And we'll do this by considering a very small simulated toy single-cell RNA-seq data set, which we've simulated with the splatter framework. This data set, which consists of 300 cells and 500 genes, has its cells split equally between three groups. The group in turquoise represents the background data set, or an uninteresting control group, and the two other groups in purple and in green represent our target data set, the groups that we're actually interested in differentiating. Obviously, there's this big batch effect that's separating these groups, and so we would like to try and mitigate the effect of this batch effect using some kind of dimensionality reduction method. That's what we'll do in the coming slides. So the first two dimensionality reduction methods that we're going to consider are TSNA and UMAP. And unsurprisingly, they're unable to handle this batch effect. They really don't have the tools necessary to do that anyways. And so this is kind of more of a sanity check. However, when you look at our contrastive methods, CPCA and SCPCA, we immediately tell that by harnessing the unwanted variation in our control group, we're able to almost completely remove our unwanted variation in the target data. Now, if you look carefully at CPCA, or the CPCA embedding, you'll see that this unwanted variation is not completely removed. In fact, you could still see a stratification of the batches on the second contrastive principal component. If we instead turn our attention to the sparse contrastive principal component analysis embedding, we find that the observations of different batches are actually completely mixed within each biological group. And so this extra stage of sparsification that SCPCA performs in addition to the contrastive procedure removes any remaining technical effect. So clearly SCPCA outperforms the other more general dimensionality reduction methods in this simplistic setting, but we also want to compare it to state-of-the-art methods for single-cell RNA-seq data dimensionality reduction, in particular ZenBWave. And so now we consider two settings of ZenBWave, one in which this batch effect is considered known, and one in which it's considered unknown, an unknown confounder. And we find that when this batch effect is unknown, ZenBWave performs similarly to TSNA and UMAP. And when it is known, ZenBWave successfully regresses out the batch effect in this low-dimensional embedding. However, you'll still notice that the clusters are not as nicely separated or nicely formed as the SCPCA embeddings. And that's it. That's all I have time to show you today. But if you found that this method was interesting, then I encourage you to go check out our paper or to review our packaging. Thank you.
All right, wonderful. Thank you all four speakers for very interesting talks. Keep the questions coming in the Pathable poll. So the first question is for you, Wang. And the question is, how do you sort the gene expression? Because in different samples, the expression level of a gene can be different.
Hi, thank you for this question. It's a very good question. We normalize the genes using RPKM because there is some length bias also cause some extra noise or confounding effects to the correlation. So we use the RPKM to normalize and then use the average RPKM across all the samples, which is the average RPKM of each gene to sort the correlation matrix. Thank you.
All right. The next question was for Ting Huang. Why do converters to max quant or OpenMS do, and why are they needed?
Hi, thanks for the question. MSstatsTMT so the main reason is MSstatsTMT use the output of data processing tools as input. The data processing tool produce a list of peptide spectra matching with reporter intensities, but different processing tools such as MaxQuant, OpenMS, Proteome Discover, they really have very different output format. Some may be text, some may be Excel, and some may be long format, some may be short wide format. So our MSstatsTMT package, we have a uniform format required as input. So in order to help our user use MSstatsTMT, we provide the converters for different data processing tools. For example, when you run the data processing tool, you can directly output have their output files and run MSstatsTMT without any further data manipulation. Inside the converter, the most important thing, it will produce the required input format, but also inside the converter, we will also pre-clean the data. It will do some filtering, and if there's a fractionation, the converter can also combine the fractions.
All right, great. So the next question is for Simina, and the top polling question was, why is it better to add the covariates or metadata at the correction step and not directly into the model?
So that's a really good question. So one issue one sort of obvious thing, right, is like you might just have the p-values. And so of course it's always better to have the raw data, but in some cases you don't. It's very common for GWAS studies that you only have the p-values or maybe like a summary of the model coefficients for each SNP because the data sets are very large and also because of privacy issues. Whenever you're looking at germline data, that's always a problem. And then the other issue is that it doesn't really make sense to add them at the stage of the model in many of these cases because the covariates are actually for the feature. So for example, for the GWAS example, if you have, the sample size is a feature of the study for the specific SNP, right? Same with the minor allele frequency. So you can't really fit a model for the SNP and have those variables in because they're just going to be constant, right? So in some ways, I think it's more similar to the meta regression case. So for meta-analysis, you combine a lot of studies that address the same question. For meta regression, you can add an additional variable, which is like a study level variable, for example, like the year that a study is published in or the kind of study. So I think that's sort of the basic idea. That's something that we're actually looking into these connections.
Great, thank you. So the next question is for Philippe. How do you ensure the stability of the PCs? Kind of a related question is, how many PCs are you applying the regularization to?
So we apply the SPCA algorithm essentially to the contrastive covariance matrix you get from doing the difference of your target and background data set matrices. And so by doing that, you essentially get to only keep maybe like on average leading 10 principal components or contrastive principal components. And then, so if you want to run SPCA, you'd have to run it on only those 10, but you could even reduce it further and only use, let's say, the top 2 or 3, if that's what you plan on plotting. And that's controlled via the SCPCA package.
All right, thank you. So jumping back to our first speaker again, is the PCA, this is related specifically to the last slide you were showing, is the PCA here used just for denoising? Is there a threshold for the number of PCs that you have been able to calibrate at your end across multiple data sets that you can suggest for a better signal?
you for this question. Actually, I think it's still an open question to choosing the number of PCs. And the removing PCs is mostly for removing the battery effect in the network analysis. And compared with SV and comeback, this method don't need the knowledge of the battery effect information. And in the paper that created this method, it suggests to use the SVA package to estimate the surrogate variables as the number of principal components, and it shows that it remove this number of principal components is better than removing like half of the number of principal components, but we didn't do actual analysis here, and we also tried removing 4 PCs because in this number of PCs we can see a centered distribution of correlations of all the correlation of the entire correlation matrix, and we also still see a positive shift of the ribosome genes, which suggests that the 4 PC is also a good choice, but overall it's still an open question. Thank you.
Thank you. So the next question is for Simina. For your GWAS example, the p-value of significance that was shown was p less than 0.05. Typically for GWAS, significance is much lower, 5 times 10 to the negative 8, for example. Does your package consider the multiple testing in such a way that the GWAS significance can be larger?
Yeah, so I mean, we were just, I think maybe this refers to the summary slide where we were comparing how many SNPs are called significant at p less than 0.05, or the FDR, which you could think of it as like an adjusted Q value at Q less than 0.05. So we definitely don't recommend doing any analysis that doesn't consider multiple testing. So that was just, I mean, you can just see, of course, that with our method you have a lot fewer than if you just look at the p values. So it's true. So in general, for GWAS, in the GWAS literature, for several reasons, like the fact that you expect a very small number of SNPs to be truly associated with the outcome, and I think also that you can get a really large sample size, it's more common for people to consider the family-wise error rate versus the false discovery rate, and they consider like a genome-wide significance rate, which is like the 5 times 10 to negative 8. Which isn't quite Bonferroni corrected, but it adjusts for, I think it just adjusts for like correlations. There's some issues with it because I think the correlations are based on European data and linkage disequilibriums, but so that is going to be a stricter correction. So we're just, in our case, we're considering the false discovery rate and not the family-wise error rate.
Thank you. So Philippe, for CPCA and SCPCA, do the numeric axes have meaning similar to PCA, and can they be used as covariates in the model? This is kind of in contrast to TSNE and UMAP, where the axis numbers don't have kind of a meaning.
Yeah, I really like this question. So if you ignore for a second the hyperparameters in CPCA and SCPCA, what it's really doing is an automated pruning of the leading principal components that are typically only associated with some kind of uninteresting variation. And so the remaining components that are left in both CPCA and SCPCA can be thought of as just like regular PCA components. And so that means you could include them in a model. We haven't actually done that yet, but I'd be super interested if you had any ideas on what to try it out and to discuss it more. Hopefully that answers the question.
Wonderful. So yeah, so maybe this is a good time to mention that there are a lot of ways within the conference platform to kind of continue discussions with the speakers. There's no way we're going to get through all the questions that we got in this session. So, please reach out in Slack or through some of the pathable communication functions if we don't get to your question, or if you want to follow up and discuss further. So the next question is for Yi. It's, would your method work for VST normalized data as well? You mentioned RPKM because it accounts for gene length. But in the case of, for example, WGCNA, comparing correlation of the same gene across samples and not within the sample, this wouldn't really take gene length into account. Hopefully that's clear.
I hope I understand the question well. So actually, our methods can apply for correlation metrics that comes from the expression level normalized by anyways. As long as we have a correlation matrix, we can directly apply our method. And I think for WGCNA, it requires the input of the correlation matrix and then estimate some clusters. And we also tried out, we can also, our method also compatible with WGCNA because our work only applies on the correlation matrix, and anything up before the correlation matrix can be compatible with our method. Thank you.
Wonderful. Thank you. All right, so the next question is for Simina. Is it possible that the way that you fit the logistic regression overfits to the data and provides inflated adjusted p-values?
Yes, I mean, overfitting is always a possibility. I mean, that's why we're very happy with the results that that we got, that Keegan Kortar and Stephanie Hicks and their collaborators got in the benchmarking papers. I didn't go into a lot of details. I didn't go into any details in the talk about how we're not doing this at just one threshold for the p-values. We're actually binarizing the p-values at a bunch of different thresholds and then getting a smooth estimate. And that's basically what is done for the q-value package and method as well. And that's described in our paper if you want more details. I think it is true in general that, I mean, we actually also talk about this in our paper. I mean, adaptive methods in general are going to work better if you have a data set with a lot of features, but that's usually when you're more interested, I think, in trying to get more power. So if you have, maybe just like 1,000 features, you might end up maybe coming up with something that's more likely to be anti-conservative than if you have, like 20,000 features. And again, this is an issue with the q-value methods. And, if you want to be very conservative, you can go with Benjamini-Hochberg, but then there's going to be this trade-off. But then if it's if it's a small data set, maybe that's, maybe you're better off with that.
Wonderful, thank you. Well, that's the end of the time we have. I'd like to thank all 4 speakers one more time. Again, please keep conversations going in any of the available communication channels, and thank you all.